My name's Andrew Cowley from the European Bioinformatics Institute, and I'm going to give a short webinar on using similarity search tools at EMBL-EBI. So the first question people often have is, well, why bother with similarity searching? Why is this useful in the first place? Well, it's something that's been going on for a long time in bioinformatics now, and it's now become central to genome annotation, to help us classify proteins, and to explore distant evolutionary relationships. How do we do that? Well, we start off with a query sequence. This could be an unknown sequence or a sequence that we want to investigate. And we align that sequence with other sequences in the EMBL-EBI databases. During that alignment, we can assess the similarity, and through that, we can infer homology, which is shared ancestry. And it's this homology inference that allows us to transfer information and make use of what we found in the database. So I've got a question for you to keep in mind as we go through this webinar. Are you happy to be wrong one time in 1,000? So just keep that in mind as you go through the seminar. I'm going to start off by showing you how to find our sequence similarity search tools at EMBL-EBI. Our website is www.ebi.ac.uk. And this is what our front page looks like. The first way that you can find search tools at EMBL-EBI is using the EBI search box. This is the big search box in the middle of the home page, and you can just type in the name of the tool that you're looking for. You can also use this to type in the name of a gene or a protein of interest, and it will search all our databases for it as well. Another way of finding the search tools is to browse the services that are available. You can do that by going to the home page again, and then clicking on any of the links to the services. We have one down here, and we have one at the top of the page as well. Going to the services page shows the different categories of bioinformatics services that we have at the European Bioinformatics Institute. And you can find the sequence similarity search tools in the other software link, and also in the DNA and RNA and the proteins link. You can also go directly to a menu page for sequence similarity search tools, and there's a direct URL for that, which is www.ebi.ac.uk/tools with a capital T /SSS. So if you're following along now and you want to follow along and look at this with your own browser, then I suggest you open a page and go to that link. That's www.ebi.ac.uk/tools with a capital T /SSS. And this page looks like this. So there's a menu here of different sequence similarity search tools. And you open the input forms for these tools by clicking on the links underneath each tool. So if I was going to look at protein sequence searching, so I want to search the protein sequence databases at EMBL-EBI, I would click on the protein link under, for example, the NCBI BLAST paragraph. So the next question might be, which tool do I want to use? There's quite a range of tools available. So here are my own recommendations for tools. If you're looking for the fastest search tool that we have, or you're only interested in finding the most highly similar sequences, then go ahead and use BLAST. It's very, very quick. If you need more accurate statistics as the result of your search, then I recommend the FASTA suite of tools. And for a single run, our most accurate local alignment search tool is a tool called S-Search. Some more recommendations. Say you have a short query sequence, or you need to match all of your query sequence, then I recommend our global alignment search tools. These are GG Search, which searches for global in the query and also all of the database sequence, or GL Search. This is global in the query sequence, but a local alignment in the database sequence. If you need speed, but you want to do an iterative search strategy to create a custom scorer matrix, then I recommend BLAST. On the other hand, if you want our most accurate tool that's the least prone to errors, then I really recommend PSI-Search, and we'll look at that one a bit more in detail later. So right now we're going to go ahead and perform a BLAST search just to show you what the page looks like. If you're following along, then you can click on the protein link under the NCBI BLAST section, or you can go directly to the URL www.ebi.ac.uk/tools/SSS/NCBI BLAST. I'll say that again. That's www.ebi.ac.uk/tools with a capital T /SSS/NCBI BLAST, all one word. And when you get there, you'll see a page that looks something like this. This is our input form for most of the tools that we have at EBI, and hopefully it will look quite similar between different tools. And it's split into different sections according to which step of the process you're looking at. So at the top we have the database selection, then we have the sequence input, then we have a section to allow you to change the parameters, and finally there's the button to submit the job, to run the job in the first place and carry out that search. So let's look at those in a bit more detail. At the top we have the database selection. So here's the fairly obvious box where you choose which databases you're going to be searching with the tool like BLAST. And what you do is you check or you uncheck the different boxes in the window. You can scroll down to see more options, and you can also expand some options. If there's a triangle, you can click on the triangle to expand or contract the list of sections. So there may be some subsections for databases which you can select. You can select more than one database if you like, and you can also clear the selection. If you're going into the protein section, but you've changed your mind and you want to search for nucleotide databases or for vector databases, you can click on the links at the top of the page to do this. Next question perhaps, which databases should I search? Well, for homology searches, which is the most common kind of searches, my recommendation would be to search the smallest database that you can. So if there's a database which contains your sequence of interest or likely to contain the hits of interest, go for the smallest one you can find. This helps the real matches to rise above the statistical noise, so you're more likely to find them and you're more likely to be correct in assigning homology to them. You can also use taxonomic divisions to cut down the search space. This is especially true if there are some well annotated model organisms of interest to you. If you're looking to transfer information from sequences in our database to your sequence, then it makes sense to search databases that have well curated entries and are really enriched with extra information. On the other hand, if you want to look for a comprehensive search, or you're looking to look up to see if we have something in our database, or you're looking for prior art, for example, then we have a number of comprehensive databases as well. But do be aware when you're selecting these, they may be redundant, so you may have more than one hit for the same protein, for example. And you can also add in redundancy when you're selecting the databases, because we allow you to select more than one. So just be careful that you're not selecting, say, all of UniProt plus UniProt, the SwissProt section of UniProt, or the Tremble section of UniProt, because you're selecting the same data twice. Now, behind the scenes, we try and deal with that, so it shouldn't matter too much, but it does look a bit silly if you then reference your search and you say that you searched the same bit of data twice. Moving down the page, the next section is where you upload your sequence. So you can either upload this as a file, or you can copy-paste it. And we'd like the data to be in a supported sequence format. I'll talk about that in just a second. There's one final way of inputting data to the forms, and that's using this DB:ID shorthand. And that's for searching with an entry that you already know exists in one of our databases. And it's just a shortcode, so we don't have to input the whole sequence or the sequence entry. We can just type that shortcode in, and the tool will fetch the information from the database for us. So I talked about sequence formats. Now, unfortunately, different databases store sequence data and information about sequences in different formats. So here we have two very common examples. Now this is the same biological entry, but described in EMBL format and described in GenBank format. And you can just see from the overview there, they look a little bit different already. And we can zoom in a bit further. So looking just at the top of those files, we can see in EMBL format, they describe the accession using the code AC, whereas in the GenBank format, they use the whole word accession. Even looking at the way that they represent the sequences is different as well. So we can see in EMBL format, they start the sequence with the SQ line, and then they present the sequence, and they have a number character at the end saying where in the sequence they're up to, and it's at the end of the line. In contrast, GenBank starts with origin in this case, and the sequence numbering begins at the beginning of the line. So you can see that if you used a computer to read these formats without taking into account the differences between them, you can cause problems. And there's sometimes a bit of confusion as to which tools take which formats. Now we try to make our tools take all of the sequence formats that we know of and think are good formats, but if you're looking at different tools, there's one other format I would recommend you have a look at, and that's the FASTA format. And here we have a single line at the top of the file containing the identifier and other useful information. And then on the line below that, we have the sequence information. And that's all there is to this file. It's a very simple file. It tells the program where it can find the start of new sequences, so you can have multiple sequences, and it's much easier for computers to understand. It's also a bit more compact, so you can have larger sequences for a smaller file size. But what I'd really ask you to do when you're using tools that deal with sequences is not to use Microsoft Word. .doc is not a format that many tools recognize. It's not a known sequence format. There's no real information here about where sequences start or stop, and you might have a new line. Does that mean it's a new sequence? Or does it mean that your sequence has just wrapped around and is starting a new line? So our tools and computers are not brilliantly clever, and they can't really cope with sort of Word formats like doc or Excel and things like that. So do try and make sure your data is in a known format, in a plain text format ideally, and I really do recommend you have a look at FASTA format for that. If you want to convert your sequences from one format to another, we do have some tools available at www.ebi.ac.uk/tools/SFC, that stands for Sequence Format Conversion. And one of those tools, for example, is Emboss Secret. And it's a really simple to use tool that's very good at converting between different formats. But please do remember, the tool can't add information where there's no information in the first place. So if you're going from a lightweight format into a format that has lots of description fields, but you don't supply any extra information, it won't know what to fill out those fields with, so they'll remain empty. So it can't work miracles, unfortunately, but it's still quite a useful tool. Okay, so the next step down the input form is the parameters selection. Now we try to make sure that the tools are set up with some useful parameters, so useful defaults, sorry, so that you don't have to worry too much about the parameter selection for the usual kinds of uses of these tools. But if you want to view them, or in particular if you want to change the parameters, there's a button you can click called More Options, and doing so expands out the box and shows you the different parameters that can be set for each tool. Each of these parameters has a link to some small amount of help documentation as well for it. The final step on our input page is the submission step. So once we've got everything else okay, we've got the input, and we've got the parameters set, then if you want to run it in your browser, you can just click the submit button. You can also tick the email box, be notified by email box at the bottom of the form, and this expands out, gives you a box to enter your email address, and that allows you to be sent an email containing a link to the results when your search is finished. It won't contain the results in the email, it will just contain the link back to the page, but that means you can close your browser and you don't have to worry about losing the results. So if you're following along and you want to try this yourself, then let's try a quick BLAST search now. If you're on the BLAST page, fine. Otherwise, don't forget you can go to it, www.ebi.ac.uk/tools/SSS/NCBI BLAST. Once you're there, I'd like you to change the dataset, the database, to uniprot-kb/swissprot. So this is the swissprot database. It's a smaller, highly curated database. And for the input, we're going to type in the DB ID uniprot-kb:/WAP_rat. That's uniprot-kb:/WAP_rat. So you can just type that into the input box, and that tells the program to go and fetch the sequence from our database in the uniprot database. And then I'd like you to press the submit button if you're following along. On the page, that looks like this. So don't forget to untick the first box, the uniprot knowledge base, and check the second box, uniprot-kb swissprot. And now you can see I've typed in uniprot-kb:/WAP_rat. So after that's run, your input will be checked for some simple errors, and assuming your servers aren't too busy, you'll get a result fairly quickly. And the result will look something like this. This is a result I did a few days ago, and straight away it opens on our summary table, giving you lots of information. So we're going to go through the different steps, different parts of this page now. At the top of the page, we have some navigation tools. So we have some links back to the tools menu if you want to change the tool you're using. The next line underneath tells me the job identifier. This is useful because it means you can send this to other people. You could send the URL to other people as well if you wanted to share the results. And underneath that, we have a list of results tabs. This is our way of navigating through the different pages that are available for the results. On the left of the screen, we have some selection tools. And then we have some other options which apply to those selections. So we have a button that allows us to show or hide the database annotations. We have a button to show the alignment between the query and the database. We have a button that allows you to download the selected hits in, for example, FASTA format, or perhaps the complete entries. And then we have a button to send the results to another tool. So you might have performed a BLAST search, found some sequences, and now you're interested in performing a multiple sequence alignment using something like Clustable Omega with the selected sequences. So you can use this button to quickly launch a multiple sequence alignment from the results of your BLAST search. In the middle of the page is the summary table results, and these are the hits of the database where we found alignments with your query sequence. On the left, again, we've got the selection tools. The next column along shows the DB IDs. If you remember, we used that to do the search in the first place. The next column along tells us some information about the database hits. And as well as that, it also contains links to other EBI databases that reference this hit. So for example, if you found a particular protein, we might also have other databases that talk about this protein. We might have a database which contains the nucleotide sequence entry which encodes for the protein. We might have a structure for the protein. We might have literature references in it. And so we try and collate all of those useful bits of information and give you links out to them. They're called cross-references. And we give you links to them under each hit. So you can quite quickly navigate out from a search result into other databases of the EBI and other pages that contain more information about different aspects of the same hit that you've got. Moving along, we have some results of the alignment. We have the length of the database hit. We have the score of the alignment. We have the percentage of identical residues or bases. And we have percentage of similarity residues or bases. And then finally, we have the all-important E-value. So what is this E-value? Well, it stands for expectation value or expectation score. And it's a measure of the chance of that alignment occurring randomly. Or another way of putting it is, how likely am I going to be wrong in assigning homology as a result of that hit? And remember, that was what we were trying to do in the first place. So low values are good. We want to be wrong less often. We want to have a low chance of being wrong. And a very common cutoff is 1 times 10 to the minus 3, which is the same thing as saying, I'm happy to be wrong 1 time in 1,000. And a quick note about the way that this is represented on the page. On our results, we use 1e minus 3. This is saying the same thing as 1 times 10 to the minus 3, which is the same thing as 0.001. So it's just represented in different ways, but it means the same thing. So what are the main factors that affect e-value? Well, as well as just the alignment, the length of the sequence affects the alignment. So small length alignments are more likely to occur by chance. The other one is database size. If you have a bigger database containing more entries, then there are more occasions to find that alignment by chance. So both of those result in a worse or higher e-value score. Looking at our other tabs now, there is a tool output tab, which just shows you the text output of the program, so I'm not showing that now. And then the next tab along is called the visual output. And this shows you where the alignment is occurring in your sequences. So a tool like BLAST is a local alignment tool, so it might not align across the whole length of your sequence. And this page shows you which region in your sequence the alignment has been found. And on the left-hand column, we have the region of the query sequence that's been found as aligning, and on the right-hand column we have the region of the database sequence that's aligning. On the very left we have a control which changes the coloring scheme between either a dynamic scheme where the colors are shaded according to the range of e-values in your results, or a fixed scheme which sets them fixed according to fixed values. And at the bottom of the page, it's not shown here, but at the bottom of the page there's a little color scale so you can correlate that with e-values. So this is quite useful if you found a match and you want to check the size of the database sequence that you've matched. If you've matched all of your query sequence, but only a very small portion of the database sequence, then you need to be a little bit cautious before you say that those two sequences are doing the same thing, perhaps. There's obviously a significant alignment in that portion of the sequence, but if some annotation you're looking at is related to a different part of the sequence, you need to be careful about transferring that. The next tab along is a really useful tab called Functional Predictions. And this tab contains domain and family predictions from Interpro for all of the hits that you found in the databases. So again, we have an e-value coloring button on the left, which does the same thing as on the previous tab. And then underneath that, we have a little control which allows us to select which Interpro databases we're going to be showing the results for. And I'm just going to zoom in now to the result to show that a little more in detail. So we've just zoomed in, and the first thing to note is the color shaded box, and this is the alignment region. So the very top line, we have our query sequence, and the line underneath that shows our database sequence, and the shaded box shows the area of the alignment. So far that matches up with the previous tab. But underneath that, we've got the regions of the database hit that are found to match Interpro families and descriptions and motifs. So you can see the shaded regions underneath that show the domain and family regions and where they're occurring in the database sequence. So before you transfer annotation and particularly functional information from your database sequence to your query sequence, you really want to check that your alignment also covers those same domains that are responsible for that function. So Interpro is a really good resource for checking the protein domains, which are very functional. And there's a list of the descriptions on the right-hand side as well for the hits that it's found. So yeah, this is a really good tool to check the accuracy of you transferring information, and to also check that you're not aligning in a domain which is unrelated to your protein. So there are some errors that sometimes occur in alignments which can result in the alignment stretching over unrelated domains. And this display will help you to see where that happens. Moving on, the next tab is called the results summary. And this just lists the available results that we've got from your search. So there's lots of different types of BLAST formats, for example, that can be downloaded. Finally, we have the Submission Details tab. And this shows you lots of details about the job that's just been run on our servers. So it tells you the name of the program that was run, the exact version of the program that was run, the database that it was run against, when it was run, obviously. There's a copy of the input sequence as far as our servers saw it. So if you've inputted a sequence and the format wasn't quite right, it's worth checking here as to whether the program's understood it correctly. There's the raw output as well. And beneath that, there's the command line. This can be handy if you're running this job on your own computer and you want to recreate it. And underneath that, we then have the input parameters as well. So you always have a record of what parameters went with which job. So just some quick ways of checking your results. Don't rely on just the e-value. It's definitely the best measure of determining whether your protein or your DNA sequence is homologous with a hit in the database. But don't rely on it entirely. Do look at other information as well, especially the domains, if they're proteins, and look at the descriptions and annotations. Also, you can perform a reverse search. So this is where you take a hit in your database that's been found, and you do a search with that sequence again. Then you can look at the types of results that you're finding and see if they still match the same descriptions and whether they find similar sequences to the sequence you've just searched with as well. So far we've just carried out a single run search. I'm now going to demonstrate how to perform an iterative search. So single run searches, like the BLAST one we've just carried out, use a preset scoring matrix such as Blossom 62 or PAM 250. And these contain scores for different amino acid substitutions. So it says each time I encounter a cysteine and I'm matching against a cysteine in my alignment, how much score am I going to give that for the alignment? And that allows us to say that some substitutions are more likely to occur than others. So it's quite a useful way of making the alignment more biologically relevant and accurate. The problem with these is that they apply the same scores across the whole of the sequence, regardless of whether that position is conserved or whether it's variable. So here I have a pileup of several different sequences, and we can see that some positions are more conserved than others. What iterative searches allow you to do is to build a custom scoring matrix from the results of a normal search. This results in a position-specific score matrix, or PSSM. This allows the alignment to be worse in variable regions without affecting the score, whilst also saying that in conserved regions, conserved positions, I'm going to be a bit more rigid about my scoring. So just underneath we've got another picture zoomed in showing some conserved positions of a particular alignment. By doing this, by allowing variability in regions that are not so conserved, but conserving where we can, where we need to, we get an increase in the sensitivity of our search. So we can find homologs that are more distantly related. We have several iterative search methods at EBI. We have Sci and PHI-BLAST, and we also have PSI-Search. So if you're following along and you want to try this again, then you can go back to the Tools menu and click on the protein link under PSI-Search, or you can go directly to www.ebi.ac.uk/tools/SSS/PSI-Search, all one word. So have a look there and make your way to that page if you're following along. When you get to that page, you should hopefully see it looks fairly similar to our other tools, but there's two main differences from the BLAST search. The first one is that the database selection is a single drop-down. There's no opportunity here to select multiple databases. The second difference is we have something called a PSSM e-value cutoff And I'll explain what that does in a slide or two. But you can again leave that at default, and then you do the same entries that you would normally do. So if you're following along, then again I recommend changing the database to uniprot kb swissprot. Type in the DB ID of uniprot kb :wap_rat, and then press the submit button. Now searches with PSI-Search are a little bit slower. Before I say that, here's a screenshot of what that input would look like. So you can see we have a drop-down menu now for the database selection. Okay, sorry, I was saying PSI-Search is a little bit slower than BLAST because it's actually made up of several differences. PSI-Search is using at the base of its tool the S-Search program. So from that list of recommendations I gave earlier, I said S-Search is a very accurate search tool. So already it starts off being very accurate, but it is a bit slower than a heuristics tool like BLAST. So we start off accurate local alignment search tool. The next thing we add to it is that iterative generation of a PSSM. So that, like PSI-BLAST, helps us to add sensitivity. And the final step in PSI-Search is something called HOE masking, homologous overextension masking. I won't go into the details of that in this talk, but what that does is helps to reduce errors. So that improves the selectivity of our results as well. So back to what that cutoff threshold has done. What it's saying is I'm going to pre-select a number of my results from the first run, and those results I'm going to generate a new scoring matrix from, and that's the PSSM. So I can manually change that selection. All that parameter did was tell me which ones are pre-ticked. If I was to scroll down, I would see that there are ticks all the way down for the results up to 10 to the minus 3, 1 times 10 to the minus 3 expect score, and then underneath I'd find them unticked. But I can manually change that as much as I like. When I'm happy with the selection of the results that I'm going to use to create my new, more sensitive PSSM, then I click the Run Next iteration. So if you're following along, go ahead and do that now. Okay, so after some time, hopefully, you'll be able to get at the results from your first iterative run. So that first search we ran was just setting up the PSSM, and now this is the result of the first search using our custom PSSM. The first difference in the results is that we have this word saying new or old. So that's saying whether a sequence was significant in our last round or not. What you'll find with PSI-Search is that as the iterations run on and the sensitivity improves, when you go down, further down the results, you'll hopefully finding more results that are significant hits now, and these will be flagged with a new each time they're new. And that shows you that these results weren't considered significant before, but now with the new improved PSSM, we're finding them. So it's a result of the improved sensitivity. There are some extra buttons as well. You can view the threshold limits. You can jump right down to your 10 to the minus 3 cutoff. You can save the progress and come back to it later. Sometimes you've run several iterations. And you can also download the custom PSSM that gave the results you're seeing. These PSSMs are themselves some useful information sometimes, telling you the frequency of different substitutions in your results. Okay, so those web browser searches are fine, but 1 or 2, yeah, I can do 1 or 2 through a website, fine. But what if I want to run 20,000 searches? So for that, we have something called programmatic access via web services. And these are APIs using things like SOAP and REST, and they allow you to programmatically run searches or other tools against the data in our databases. And it's all run on our servers, so you don't have to worry about the compute power, you don't have to worry about storing these big databases or keeping them up to date, but you can run scripts against them as well, so you can automate or run through lots and lots of analyses systematically. We have pre-compiled sample clients, so you can just download those in several different languages. they're as simple as downloading them and running them in most cases. And there's some more documentation and the links available via the Web Services button at the top of the tools pages. If you'd like more documentation and help, then that's available through the Help and Documentation link at the top of the pages. We also have a help desk that's available via the feedback button on the right-hand side of the screens, or through the link or the URL www.ebi.ac.uk/support. And if you have access to journals, then there's a really nice paper that describes in a step-by-step fashion how to use several tools and several command line versions of web services in current protocols in bioinformatics, Unit 3.12. And that's called Using EMBL-EBI Services via Web Interface and Programmatically via Web Services. It's a nice, easy to follow unit. If you do find our tools useful, we'd be really, really grateful if you could cite them. And this is the URL, this is the paper reference for citing the use of tools in that framework, which is the EMBL-EBI Bioinformatics Web and Programmatic Tools Framework. And that's a nucleic acids res web server issue. And there's a DOI there for you to follow. Citations are really, really important, and they help us justify our work on our tools. So in summary, don't just use the same old tool. Think about what tool you'd like to use. Think about choosing the right database, particularly choosing small databases. They really are good. And do some simple checks to see if the results make sense. And just a final thought to leave you with is that sometimes it's okay to be wrong more than one time in 1,000, because that's just the e-value score. And we know that you'll probably still be able to find homologous sequences with a lower score than that, but then you're going to have to start checking. You can't assume it. So thank you very much for watching. I've just left this slide here with some more links, and we'll leave you for a few minutes to compose some questions if you like, if you're watching this live. If you're not watching this live, please do get in touch via the support link, or you can email me about the contents of this talk at andrew.cowley@ebi.ac.uk, or you can contact our help desk through the email support@ebi.ac.uk.